Glycolysis is a series of reactions that ultimately splits glucose into pyruvate. Glycolysis occurs in almost all living things, serving as a starting point for fermentation or cellular respiration. There are two phases of glycolysis. The first phase is energy investment phase. Hexokinase transfers a phosphate group from ATP to glucose, making it more chemically reactive. The charge of the phosphate group also traps the sugar into the cell. Then the glucose 6-phosphate is converted to fructose 6-phosphate by rearranging covalent bonds. Phosphofructokinase transfers a phosphate group of ATP to the opposite end of a sugar, investing a second molecule of ATP. This is a key step in regulation of glycolysis. Aldolase cleaves the sugar molecule into two different 3-carbon sugars. Conversion between DHAP and G3P, the reaction never reaches equilibrium. G3P is used in the next step as fast as it forms. This is the beginning of the second phase, the energy payoff phase. Two conceptual reactions. First, sugar is oxidized by the transfer of electron NAD positive, forming NADH. Second, using energy from exergonic redox reaction, a phosphate group is attached to the oxidized substrate, making a high-energy product. The phosphate group is then transferred to ADP, which is a substrate-level phosphorylation in an exergonic reaction. The carboxyl group of G3P has been oxidized to the carboxyl group of an organic acid, or 3-phosphoroglycerate. This enzyme relocates the remaining phosphate group. Then enolase causes a double bond to form in the substrate by extracting a water molecule, yielding phosphorinolipyruvate, a compound with a very high potential energy. And at last, the pyruvate first enters the mitochondria through an active transport with a transport protein. Then its carboxyl group is removed and released as CO2. The remaining 2-carbon compound is then oxidized in order to form an acetate, where the electrons taken are transferred to NAD positive, creating NADH. Then the sulfur atom of the coenzyme A attaches to the acetate, creating a product of high potential energy, acetyl-CoA. This product is used later in the citric acid cycle when the mitochondrial matrix.